Hi, good afternoon and welcome everyone. I'm Pat Furlong from Parent Project Muscular Dystrophy and I'm joined by my colleague Annie Kennedy as well. Today we're going to give the community an update and we're thankful for Akashi Therapeutics to come online and be able to tell us about the new developments and the ability to restart the HT100 study. As we all know, we lost a young man some time back with a new study and it was really a crushing blow for our community and for Akashi. So after a whole year and more of investigation, the FDA has lifted the clinical hold and HT100 will be back in clinical study. So today we're going to learn about what was done, what Akashi has learned, and what their plans are for the future. Online from Akashi is Marc Blaustein, Diana Escolar, Chief Medical Officer, and Ernie Bush, the Chief Scientific Officer, and Marc, as you know, is the CEO. So Marc, I'm going to ask you to start in, and we'll follow with questions that have come in, as well as some questions that may come up during the session. Marc?
Excellent. Thank you very much, Pat. And thank you, Annie, as well, and to PPMD for hosting this webinar. We're very excited to be taking HT100 forward in the clinic, but before we begin the webinar, I want to pause, as Pat just did, and recognize that we're here today because of a boy who's a hero to everyone in the DMD community. As Pat just mentioned, a boy died while bravely participating in a clinical trial of HT100, and I know his family, particularly, and of course the whole community, feels that loss every day. Those boys and young men and families who participate in clinical research do so because it's necessary for the greater good of the community, and so we can have safe and effective treatments for all who have DMD. So, speaking on behalf of the entire Akashi team, we're engaged in this effort because we want to find treatments that will make a positive difference for all boys and young men with DMD. And this death was the most devastating possible outcome for us. But we want to recognize and thank this child and his family and honor him by continuing the effort to develop safe and effective treatments. So with that as a background, let's move forward. So I know many of you may not know Akashi Therapeutics. We are a clinical stage biotech company focused on DMD. We are founded by patients and led by a team of veteran industry executives with a mission to turn Duchenne from a fatal genetic disease into a chronic manageable condition. And we have a portfolio of products that together we think are going to be a really critical part of doing that and changing the face of DMD treatment in the lives of boys with DMD and young men and their families. So we have three treatments in development. The first is HT100, and we'll spend most of the webinar today talking about that. The second is a treatment called DT200. It's a muscle builder that increases muscle mass and strength, and that is also a clinical stage candidate. And then our third candidate is one that we call HT300. It's derived from the venom of a Chilean rose tarantula, hence the picture here. And it's a synthetic peptide that restores normal calcium levels in channel, and abnormal calcium is one of the key triggers for the damage that occurs in Duchenne muscle. And this product we hope to take into the clinic certainly within a year. So let's focus today, though, on HT100. The active ingredient in HT100 is a compound called halofugenone, and throughout this webinar, you'll hear me refer both to HT100 and to halofugenone, and so you understand halofugenone is the active component in it. So HT100 is an oral tablet. It's given twice a day, and it has several really important biological activities. One of the most prominent for Duchenne muscular dystrophy is it's an anti-fibrotic. It actually has been shown to reverse fibrosis within muscle. It also has important anti-inflammatory and muscle regeneration activities to help promote healthy muscle regeneration. And HT100, or halofuginone, also upregulates utrophin, which, as you know, can perform some of the functions of an absent dystrophin. So HT100 has already shown promising data, and we'll spend some time with going through that. One thing to just take away is that it's the first drug since steroids to improve baseline muscle strength, and we'll show you that data. And we also have extensive safety and tolerability data that we'll talk about. So to lay the context here, provide the context, so first we'll talk about the previous existing HT100 clinical trial program and the data that came out of that. And so here's just a quick overview. There was a series of trials that we called Halo DMD 01, 02, 03, and they flowed directly from one into the next. It was a very carefully designed protocol where a single dose was given to each cohort of 6 boys, each individual boy in the cohort, and then when all 6 had their single dose, there was a safety review just of that single dose, and if that looked positive, then the boys would continue to 7 days of dosing, another safety review, and then 30 days of dosing. There was actually a 14-day check-in there, and then a cumulative review at 30 days. And then they would move into the next study right away, which had 12 months of dosing, and then into an open continuation study that was called Halo DMD 03. And all throughout this, of course, individual patients are watched by the site and by the company as data comes in, as well as these very regular reviews. So we had 4 cohorts of ascending dose that participated in this trial. Cohort 1, the boys were on drug for between 10 months, and most of them were on for over a year and a half of continuous dosing. And there were 6 boys per cohort. Some of them, of course, didn't complete this full duration, but in the first 3 cohorts, there were a total of 16 boys who had this extended treatment. Cohort 2, about a year and a half at double the dose of exposure, and then cohort 3, most of the boys were just under a year. And cohort 4 had just begun the 30-day dosing period when the boy fell sick and dosing was stopped there. As you can see, there was a fair amount of experience for long durations of continuous dosing. So a little bit about the summary of the safety. In those first 3 cohorts, the doses were from 300 to 1,500 micrograms a day, and there was continuous exposure for 10 to 18 months. And the most common treatment-related adverse events were stomach issues, nausea, vomiting, etc., and headaches. There were a total of 307 adverse events, and this is whether they were related or not to study drug. If someone falls off their bicycle and breaks their arm, that's considered an adverse event. So there were a total of 307 adverse events. 81% of them were mild and resolved without any follow-up issues. 17% were moderate, 1% were severe. 2 of these were considered by the investigator to be possibly related to study drug, and 2 of them were considered to be not related to study drug. And then, of course, we had one event that was fatal. So we immediately began an investigation as soon as the boy fell sick. So what we found as we examined this and began a very intensive investigation was that this boy's blood level of halofuginone, the active ingredient in HT100, was about 7 times higher than it should have been and had already been measured at steady state. In other words, what should have been the maximum exposure when this event happened. So one of our first questions for the investigation was, why was this blood level so high? One of the other important things that happened in this case was that the boy had very low blood pressure and it was drug resistant, a condition called vasoplegia. And so what we wanted to know was, did HT100, was that the cause of this drug-resistant low blood pressure, or was it some other unrelated issue? And then finally, we wanted to know, before there was any serious toxicity with the drug, assuming that the drug was involved in this, are there any warning signs so that action can be taken and this could be avoided in the future? And so these were 3 really critical areas of focus for our investigation. So I think, as I mentioned a moment ago, as soon as the boy fell sick, Akashi halted all dosing in the highest dose cohort, which was cohort 4, and that was the cohort that he was part of. And then soon after, in discussion with FDA, we halted all dosing in the trial, and clinical hold was placed on the trial. So this investigation took much of 2016 after this incident occurred, very early in 2016. So major components of our investigation were a comprehensive review of all of our data and related data. So we looked at all clinical data. We have many biomarkers. We actually analyzed new biomarkers from stored samples, clinical parameters, all patients, and then we brought in some independent outside experts in areas that were relevant to this case and had them review everything to provide an independent perspective. So we also undertook additional studies ourselves as part of this investigation. So one thing was a complete panel of in vitro studies to explore whether there was a drug-drug interaction, because we had some evidence that might be a factor here, and I'll talk more about that in a moment. And our work there included the full panel of DDI studies that FDA recommends in their guidance, as well as other things. We then did a new dog study to explore this potential drug-drug interaction, and we retested the product to make sure there wasn't some issue that had not been seen before with the product. So here are some of the key results of our investigation. After an exhaustive analysis, we decided that the extremely high levels of halofuginone were most likely responsible for this drug-resistant low blood pressure that led to the hospitalization of this boy and ultimately to his death. So what we found was that drug quality and errors in dosing were not a factor. The drug itself performed and had exactly what we expected in the tablets, which were returned, and it had been dosed correctly. We also found, in looking through the data, that other patients who had taken repeat dose of an anti-nausea drug called Ondansetron seemed to have a somewhat elevated halofuginone level associated with that use of repeat dose Ondansetron. And so we really began to focus on that as a potential issue, and we did the dog study that I mentioned on the previous slide. And what we found was this study supported the conclusion that it looks like there's an interaction here. So what we see is that the blood levels of halofuginone rise after about 6 days of combination dosing, and with repeat dose ondansetron, plus what is normally a well-tolerated dose of halofuginone in dogs, this proved to be toxic. So there seemed to be some relation here, some relationship between these two. We also, of course, as I said, went back and looked at all the data, not just for this patient in Cohort 4, but for all the boys who participated in the trial, and confirmed that for the doses up to 1,500 micrograms per day in Cohorts 1 to 3, that there was a favorable safety profile on those boys who've been on drug for between 10 and 18 months. And this was based on this very detailed review of all these items listed here, laboratory values, etc., and including an exhaustive look at concomitant medications, which means other drugs or medications that are given at the same time as the boys in the trial are taking HT-100. So since we identified this apparent drug-drug interaction or potential drug-drug interaction between ondansetron and halofuginone, we were very focused on whether there were other DDIs that might be present. And so there are 569 prescription and OTC drugs and supplements that were taken by all patients concomitantly with HT100. This means that it's not 569 different agents necessarily. For example, if 15 boys were on deflazacort, that would count as 15 of the 569. But it was a huge array in many of the different boys participating in the trial of drugs. And what we saw was, with the exception of ondansetron, there was no safety signal with any of these other drugs. And these included drugs commonly found in the patient community and used by boys and young men with DMD. So deflazacort, prednisone, the bone health drugs you see listed, heart health ACE inhibitors, growth hormone. There are a number of GI drugs like ranitidine and omeprazole, as well as many vitamins and supplements, CoQ10, creatinine, etc., that we know the boys take. And with all of these 569 different incidents of many different drugs being taken, there was no safety signal. So another question that you'll remember of our 3 really critical questions that we needed to focus on because of what we saw with this boy was, are there any warning signs before there's serious toxicity? And the answer was yes. So these warning signs include a number of gastrointestinal stomach issues like nausea and vomiting, as well as headache. But what we found was it's not just one of these, but it's a combination of these that one can be on the lookout for, because obviously these things can arise individually quite frequently. And we had, as by way of example, 2 other patients in this Cohort 4 who exhibited a constellation of several of these symptoms. The drug was stopped, the symptoms resolved, and then their dosing was resumed, at least for a short time, at a lower dose, and that went well, and then we had to stop we stopped dosing in the cohort because the boy felt so sick. But it's clear that there is a warning period, a sort of prodromal syndrome we would call it, before there's serious toxicity. So how do we respond to this event and the findings of this really exhaustive investigation in order to make sure that we're doing everything we can to ensure that nothing like this ever happens again? So And some of the key findings were that clearly very high halofuginone exposures are potentially very unsafe. So the first element of the protocol design is that we're moving forward with a dose of 150 micrograms, which is 15 times lower than the previous high dose, so a much lower exposure. We found this potential interaction with Ondansetron that can increase exposure, so we're contraindicating or prohibiting the use of Ondansetron. And then, remember on the previous slide, we talked about a number of what we would call early warning signs of potential toxicity are GI or stomach issues, and these can be masked by the use of antiemetics or anti-nausea drugs. So we're contraindicating not just Ondansetron, but all antiemetics to ensure those signs and symptoms are not masked. And then finally, while we didn't see any evidence of another DDI, there is the potential for that, for another drug-drug interaction. So we're limiting the introduction of new drugs or dose changes in existing ones, and where those are required, we're very closely monitoring halofuginone blood levels to make sure that we're not seeing an issue. And so these are really key elements of taking what we learned from the investigation and how we apply it to clinical development and this research going forward to do everything we can to make sure that the boys are safe. And that's absolutely our priority. So I just want to reemphasize, again, on behalf of the Akashi team, and I can tell you, of course, working with the FDA review team, that safe administration is the highest priority of both of our organizations. And after this thorough investigation and review and implementing these learnings from that investigation, we have concluded, both organizations, that HT100 can be safely dosed under the new protocol, and that was when the clinical hold was lifted and our recent announcement from last Wednesday. So let me just, I wanted to comment here because I was just talking about FDA. I did receive an email question about seeing the full FDA review, and I wanted to mention that FDA does conduct, we know, a very comprehensive review with an internal team, but they don't share that review with us. What we see is only the outcome of their review, whether they approve continued development based on the research and investigation that we've done and the data, and then the specific protocol. So, all drugs in development and even on the market carry risk, and certainly including HT100. So an important question is, why do we think it's worth continuing to explore whether HT100 may benefit boys and young men with DMD in light of the fact that there is risk in drug development with HT100? So what I'd like to do now is share a couple of slides from that present data from that existing trial where we dosed up to 18 months that show the effects of HT100. So the headline is written out at the top of the slide. Until now, only steroids have shown an improvement from baseline in muscle strength. And HT100 in this study showed an unprecedented improvement in DMD and on top of corticosteroids. And what we mean by that is it happened that all the boys in the study were already on a stable dose of corticosteroids, and so these effects were seen on top of corticosteroids. And without going into too much detail, the left panel shows all the boys in cohort 1 to 3 who had at least 11 months of treatment. And what you see is the orange line is on HT100, and when that line goes up from the start at the left, that means there's an increase in strength, and it's compared with a control arm that, as you would expect, strength goes down over time. And importantly, on the right-hand panel, it shows the same information, but only in the very lowest dose, the two lowest dose cohorts, so the boys in the HT100 trial who were receiving low-dose HT100. And you can see, if anything, the results there were at least as strong, telling us that it looks like low-dose halofugenone HT100 has a positive effect on muscle strength.
And so, Mark, by low dose, are you talking about the 1,500 micrograms per day?
So no, that was actually that was the highest dose, and that was only in cohort 3. So this is cohorts 1 to 2. So these were doses as low as 300 micrograms per day, which were the lowest doses that we tested.
Okay.
Okay?
Yes, thanks for clarifying.
Sure, thanks for pointing that out. So the next slide is the same kind of data, but this just shows the subset of boys who participated in the trial who were non-ambulant, so older non-ambulant boys. And we understand, of course, the great need not just for treatment that are applicable for boys with any mutation, but also older non-ambulatory boys. And so we were very excited to see that strength also improved in this subset of our patient population. So again, if you look, you see that orange line after 6 to 9 months clearly starts to go up and very clearly diverges from an external control group whose strength, as you would expect, goes down over time. So we think this is very exciting to have a drug that may be relevant not just for all mutations, but for boys who are both younger and older, ambulant and non-ambulant.
And Mark, this is Annie. Can you just clarify which cohort we're looking at here?
So this is across all 3 of those cohorts, that subset of non-ambulant patients, which was 7, and they're mixed across all 3 cohorts. So we allowed, I was just going to say, we allowed both ambulant and non-ambulant boys in any cohort.
And your control is with steroids?
Yes, so this was a group that was on steroids. This was a paper that was published by Ann Connolly last year looking at a very similar group of boys.
Thank you.
Yes, you're welcome. Okay. So for Akashi, what are the next steps? So it's to move into a new study. It's designed as an open label with 150 microgram per day dose, so a lower dose. But as we saw in our previous data, we don't think we found the lowest effective dose yet because the low doses seem to be effective. So we want to explore whether a lower dose will be effective, which would give us a larger safety margin. Well,
Mark, I'm sorry, this is Annie. Can I just ask you a question about that? Was that a request, since that dose is lower than your low-dose cohort, was that a request of the FDA, or was that something exploratory that the company is doing?
So it was, FDA asked us to begin with this dose, and it's something that we're interested in exploring because we are very interested, of course, in finding the lowest effective dose.
Okay, thank you.
So, Mark, in this study, as you do this open label and collect the biomarkers and outcomes that you've chosen, are you reporting back to the FDA, and will you consider, if this doesn't show the difference that you've demonstrated in the other cohorts at higher doses, will you consider a dose-finding study of increasing that dose?
Yes, so absolutely, Pat. Very good question. So, yes, we want to explore that, and we have a whole body of data which will generate data here at this dose. We have all the data from our doses from 300 up to 1,500 micrograms per day, and if we believe that this dose isn't active, then we'll have a data package to support going up to a higher dose.
All right, and Mark, in your previous studies, when you were doing the dose escalation, you were collecting biomarkers. In addition to the outcome the physical outcomes such as elbow flexion, were you able to see any trends in the biomarkers data, or were the numbers too small to really take a look at that?
You're asking great questions. So we absolutely saw trends in some specific biomarkers of muscle fibrosis and inflammation, and those actually moved within a few months of drug beginning, and then we actually saw where we had instances where boys came off drug because there was a clinical hold, that we could see those biomarkers return back to baseline after some time. So we have the opportunity to look at those and get an early read before those efficacy outcomes as to whether we think there's a drug activity at this dose.
Great, thank you.
Yes. So while, we're going to welcome a broad population of boys, we do want to give priority first to those who've already participated in the HT-100 trials, and then we'll include other boys as well. As we did in our previous study, we're anticipating a broad population, ambulant and non-ambulant, and we'll look at pharmacokinetics or exposure, safety, tolerability, the biomarkers we just talked about, among others, and also look at drug activity.
And Mark, as you think about recruiting these young men, are you thinking, and are you including young women? That's one question. And then the second question is, how many sites are you considering?
So we are not including young women. It's just young men. And in the current protocol, we're anticipating up to 3 sites, but we haven't yet done, as you know, the clinical hold was just lifted, and we haven't yet done the site work and started to move forward. So we don't know what the final list of sites will be.
And as you think about including these patients and writing up that protocol, often as we all are so familiar with, studies started 7 years and above, and they are limited by the age of 18 often. So will you in this study be considering going either very young or into the adult population greater than 18?
This will be, what we're anticipating here is just a small study. The entry criteria will probably be very similar to our last study, so up to an age of 20, as low as 6 or 7, and then, as we said, ambulant and non-ambulant, and either on steady dose, a stable dose of steroids, or steroid naive.
Great, thank you.
Mm-hmm. And so for next steps for the company, we are in the process of, we're in active discussions with potential partners and investors to raise funds to finance development of all 3 of the drug candidates that I referenced at the beginning of the webinar. Some of you may know that we were partnered with Grunenthal. They elected to leave the partnership, actually, before we began an active dialogue with FDA. And the patient death, we believe, was certainly a factor in their decision, but there were other factors. They got a new CEO, and the head of the rare disease effort left to start his own company. So we're excited now about the opportunity to work with some new partners. And this leaves us in a position where we look at the portfolio, we look at the opportunity to make a difference in the lives of boys and young men with DMD, and we're very enthusiastic about 2017 and beyond.
Great. Thank you. Thank you so much, Mark. I want to kind of take a step back and ask you a few other questions around safety, if you don't mind.
Sure.
So I know that you've looked at the antiemetics and are worried about some of those in terms of drug-drug interactions, but I want to sort of propel us into the future, if you don't mind, and ask
Pat,
sure?
Can I just interrupt for a moment? So
sure, absolutely.
The other antiemetics, other than Ondansetron, is the only one where we've really seen what we believe is a drug-drug interaction. With the other antiemetics, we just want to make sure there's no masking of any signs of potential toxicity. So it's not a concern about a drug-drug interaction. We just don't want to cover up, mask any nausea or vomiting that may indicate a problem in the future. I just want to clarify that.
Yes, thank you, Mark. And so your protocols will be around having protocols around managing the nausea without using those, so that there's no masking of symptoms, and you'll have a protocol on suspecting around a blood pressure of a certain level as you get concerned about low blood pressures as well?
Well, there is, we anticipate if there's a change in the level of an existing drug, in other words, the dose level, or a new drug is introduced, that there will be very careful monitoring of the patient if that occurs, which will include both plasma levels and, of course, general health like blood pressure and other signs.
Right. Okay, thank you. So I know you've done an incredible investigation of the prescriptions that boys are taking now, right, to include the supplements and all of the range of drugs, growth hormone, et cetera. So in looking into the future, as I think this community is really anxious to see, is combinations. So I'm wondering if you are also thinking about working with other sponsors to understand whether or not, or if there would be some concern about the combination of HT100 to include the combination with the drugs that you're currently moving forward, as well as, for instance, the anti-myostatins, the exon skipping drugs, the stop suppression drugs, all of those drugs that seem to be really fundamental in terms of Summit's drug and those that express dystrophin or utrophin, et cetera, to make sure that we understand fully, or to the degree possible in animal studies and other studies, whether or not we should have other concerns about combining these drugs.
Absolutely. So, and I would say our thinking is exactly what you described. We are eager, both with our portfolio of three therapies, to look at combinations, and also with the many other promising therapies that other companies are developing. So, and we know we have a good model in the dog, at least, for looking at a drug-drug interaction. So that is something that we'll be very aware of and we'll look for in animal studies before we would take any combination into humans.
Great, thank you. So we've had a question come in, Mark, about does Akashi believe the drug interaction with Ondansetron is due to the drug's metabolism or due to the pharmacology around the HT3 receptor?
So we looked through all sort of common and known pathways and the mechanism of potential interactions, and did, as I think I mentioned, a really complete panel. All the studies that FDA recommends and some others that we tried as well, and we didn't find that sort of smoking gun mechanistic answer for exactly how that interaction occurs.
Okay, great. Thank you.
Mm-hmm.
All right. Well, Mark, please go on.
Well, I wanted to make sure we have some people to acknowledge, and I wanted to make sure we at least had a brief moment to do that, but I want to also answer questions and use our time well.
Well, this is Annie. Can I just ask a question around your timeline for recruitment? And it sounds like you're still finalizing some items around inclusion criteria, or maybe that's wrong. So can you just talk specifics at this point? Because we know the community's anxious and eager.
In terms of our timeline for taking this forward?
And announcing trial sites, et cetera, yes.
Yeah. So as we've discussed, we're really just back in that phase. In 30 to 60 days, we expect to know a lot more. As of now, in terms of when we expect to be dosing patients or when we anticipate, if everything stays on track, we're anticipating probably in the fourth quarter of this year that we would be dosing patients.
Thanks, Mark. And given the history and given where the FDA has requested that you start at 150 micrograms per day, are there more or recruiting or more reporting issues that you have with FDA along the way, or is this now back on the traditional path?
Well, the protocol is very carefully designed to monitor these issues that we identified and sort of highlighted on that table of changes that we've made to the protocols going forward. As long as we are at this 150 microgram dose, we are going forward and gathering information. And then if we want to increase the dose, then if we believe that's appropriate, then we'll certainly talk with FDA before making that change. The approved protocol is for 150 microgram per day dose.
For an open label?
Yes.
Okay, thank you.
Mm-hmm.
And that's not, so one question has come in about the study, and is there any study weight related? Are you taking, let's see, the new study stated to be at 150
yes.
micrograms per day. Is that the same? Is that, that's the same and has no change for weight of the patient? That was one of the questions.
Yes, and good question, and especially if someone was paying careful attention, they would see that in the previous study it was weight-based dosing. We had micrograms per kilogram, but we have had too much time to review all of our data, and what we clearly saw was that exposure is much better predicted by a flat dose than by a weight-adjusted dose. So going forward, we're going to be going with flat doses because that gives us a better prediction of what exposure will be regardless of the weight of the boy or young man.
Thank you. Thank you very much. And I just wanted to, I know Diana and Ernie are on the line as well.
Yes.
Diana, and I think this community owes all of you and certainly the family who participated such a great deal of gratitude for participating in the study and you for staying in and going through all of the years' worth of studies to ensure to the degree possible the safety of this community. So Diana and Ernie, I didn't want to miss having your voice here on this community because I think they need to hear it. We really do know that it's been difficult for all of us. Losing any young man is just incredibly painful, and for this community. And we do appreciate all you've done as well. We know that this has not been easy for you, but we do appreciate you going forward.
Thank you, Pat.
I think, Mark and Annie, do you have other questions you would like to ask? Mark, this has been really informative. We'll look forward to more information along the way and certainly to the announcement of the sites opening in fourth quarter. Annie, do you have final thoughts?
No, just thank you very much to Akashi for being here with us today.
Sure. And can I just quickly flip through our acknowledgments?
Yes, we would love that.
Thank you so much, Pat. So these 5 investigators, Ann Connolly, Catherine Wagner, Brenda Wong, Kevin Flanagan, and Craig McDonald, who generated and with their teams at their sites, generated all this research data, to the members of our pharmacovigilance team, Ann Connolly and John Lowy. And then, as many of you know, Akashi was supported for a number of years strictly by the patient community. And we received really generous support from a broad cross-section of the patient community, and the logos of the foundations that have supported us and for which we are incredibly grateful are listed here. And as Pat, you said earlier, also, of course, to the families and boys who do this very important clinical research, both for Akashi and HT-100, as well as for all the other drugs in development. So a broad thank you to the DMD community.
Well, thank you, Mark. I think just by your acknowledgments of the providers, the clinicians who are involved both in the study and the analysis of the study, and certainly from all of the foundations who've contributed, we're a community that really is driving therapies and is so anxious to see this disease and the prognosis change for everyone involved, from those just diagnosed today or being diagnosed some future day to those older adult men. I'd love to say that they're older, over the age of 20 that are here into their 20s and 30s and 40s, and we're really pleased. We do have one final question about steroids. Will the open label study include steroids or have a requirement of steroids?
So we expect that we will allow either steroid naive, in other words, no steroids, or on a stable dose of corticosteroids. Either of those conditions, boys and young men will be allowed to enter the study.
Well, it sounds like an incredibly interesting study that you'll have, that you'll be conducting, and certainly we are very hopeful for HT100 that it does exactly as we hope, that it prevents or reverses fibrosis. So thank you all for joining us. Thank you, Ernie. Thank you, Diana. Thank you, Mark, for all you do. Thanks to this community. I think we do good work together, and I know it will continue. So thank you for joining us today, and we hope to connect again soon. Thank you all.
All right. Thank you, Pat and Annie. We appreciate it.